Today my lecture is on different types of bioreactors, because in the last class I tried to discuss the reaction kinetics, and at the same time I tried to discuss that what do you mean by reactors. And again I want to repeat it, reactor means the vessels in which the reaction take place. Now here if you see that in chemical reactor is a vessel in a chemical process or plant where the chemical transformation take place to generate the desired product. Now bioreactors deals with the organism or biochemically active substances derived from other organisms. So so we can find some kind of resemblance between the reactors and the bioreactors. Bioreactors, it is purely chemical reaction, and reactor is purely chemical reaction, and bioreactors usually reaction carried out with the help of either microorganisms or the biochemically active substances that comes from the different biological sources. Now these bioreactors and unit processes that are very that can be defenses like this. This is the hardcore bioreactor that we have, and if you look at that, this is connected, this is followed by some separator, heat exchanger, utility systems, and the process control, because these are the part and parcel of the bioreactor and unit processes. So to operate the bioreactors and subsequently after the reaction is over, all these processes will be involved in this particular process. Now let me discuss about the classification of the reactors, and it can be classified on the on three category. One is mode of operations. When you call about the mode of operations, we have three type of mode of operations we have. One is batch process, another fed batch, and continuous process. Now batch process means you take the material at a time, let the reaction take place. After the reaction is over, we take it out. Fed batch means we put the raw material in a batch format. There is the input, but there is no output from the system. And after the volume reach, we stop the operation and take out the product from the reactor. And continuous system, we have continuous input and output from the reactor. Now it can be classified on the basis of geometrical configurations. As for example, we have tubular reactor. Tubular reactors is just like tubes that through the tubes that liquid that passes from one end to other end, and there is the agitated vessels, because we do the stirring inside the vessels so that just to maintain the homogeneity of the reaction mixture, and so that substrate can freely interact with each other and give the products. And so if you, and third is the contact patterns between the phases, like packed bed reactor, expanded bed reactor, fluidized bed reactor, trickling bed reactors, bubble column reactor, and the air lift reactor. There are different types. You can see that packed bed reactors where the particles they touching with each other, I will show you in the diagram. And expanded bed reactor, the particles that they will be detached from each other, and fluidized bed reactor, it will be more detached from each other, and trickling bed reactor is something similar to packed bed reactor. And then bubble column reactor, we simply bubble the air through the liquid, and air lift reactor, we have some the inner draft tube inside this bubble column reactors. Now reactor classification based on the mode of operation. This is, I told you, the batch reactor. This is most ancient, I can say that the old type of reactor, that is, and basically easy to operate, because you take the raw material at a time and let the reaction take place, and after the reaction is over, you take out the material. And India has more than 250 distilleries that we have, which produce the ethanol from cane molasses, and most of the ethanol fermentation process, if you look at early days, it is actually operated. Recently it has been some of them has been converted to the continuous process. Then we have chemical industry like dyes and inks production industry that use the batch process, and particularly the waste treatment process we largely use the this batch process. Now in the fed batch reactor is mostly used where the substrate acts as the inhibition, because inhibition is a common phenomena particularly in the biological process, because if you use more substrate in the but then it gives the inhibition to the reactors, and in case of the substrate inhibition, it is usually recommended that that fed batch reactor is usually preferable. It can be defined as the tank in which the reactants are charged initially, and limiting reactants is added continuously during the operation. No product is removable during the process. This is the main important, and the examples where it can be applied is the Baker's fermentation process, where the sugar can be converted into yeast cells, that we can increase the yeast cell production by using more substrate, because usually 1 gram of sugar can approximately produce 0.5 gram of yeast cells. So more substrate is utilized in the system, we can get more the yeast cells. And also in the pharmaceutical industry, particularly for the production of secondary metabolites. Then we have this continuous reactor, particularly we have continuous flow reactor vessel in which the reaction is fed continuously and products is withdrawn continuously from it. But here I want to point out most of the continuous reactor usually operated in the batch mode and let the reaction take place. When the reaction take place, rate of reaction is maximum, then we start feeding one end and take the products from other end. The application is the ACP production. ACP is the single cell protein production, high fructose corn syrup production, that is usually taking place by using immobilized glucose isomerase enzyme. We pass glucose as a substrate which converted to fructose and activate a sludge process, which is nothing but CSTR with cell mass recycling. And it can be schematically it can be represented like this. In the batch process, we take the substrate at a time, let the reaction take place, and then in the fed batch we keep on feeding the substrate in the batch pattern, and when the reactor volume reaches to the maximum, then we stop the operation and take out the product. In the continuous system, there is a continuous inflow. You can see the continuous inflow and continuous outflow from the reactor. Now, the reactor classification based on geometrical configuration, we have tubular reactor, like plug flow reactor. Tubular reactor looks like this. It is like a tube. You can see that liquid feed is coming in one end and product is going out in other end. Now, if you look at this reactor consists of a hollow pipe or tube through which the reactant flows, and where the reactant moves as a plug along the pipe. Now, plug flow, basically in the chemical engineering language we call it piston flow. Piston flow means when liquid flows through the pipeline, there should be some kind of velocity gradient due to the friction, wall friction effect, between the fluid and the wall. So, in the ideal flow, particularly piston flow is considered as the ideal flow where there is no friction, because that is not actually possible, but normal case we have the friction. That is why we have in the cross section we have the different velocity of the liquid. Now in case of plug flow reactor, there is no axial mixing that take place across the reactor, but there might be the radial mixing. Now this is applied for homogeneous and heterogeneous reactor and product inhibition process. That this is very important, and most of the biological process we know as the product formation take place, particularly in case of acid production process. I work with citric acid fermentation process. We find that high substances, the acid concentration has some inhibitory effect on the organism. We can give the example of acetic acid fermentation fermentation process, lactic acid fermentation process, all this has some kind of inhibition effect. Even in the ethanol fermentation process, ethanol has some kind of inhibition process. So in case of product inhibition, this flask flow reactor usually recommended. In my next lecture I shall explain you why the flask flow reactor is chosen for product inhibition. And high temperature reaction also, this flask flow reactor is usually preferable. Next is the continuous start tank reactor. This reactor is very easy to operate. The reactant and the products are continuously added and withdrawn, while the contents within the vessels are vigorously stirred using the internal agitation. So this is like this, that just we put the starter. Main purpose of putting the starter is to maintain the homogeneity of the reaction mixture. We pass the substrate one end and take the product in the other, and this is very easy to operate. So schematically this is represented like this. This is the vessel, this is the starter, and we put the feed one end and we take out the product in other end. So then we have the pack bed reactor. This is reactor configuration based on contacting pattern between the phases. I told you that phases means we have maybe presence of two phases, solid and liquid. So here in the pack bed reactor, usually it looks like this, that your particles that packed in the reactor, they touch each other due to gravitational force of attraction. The major problem of this process is that when you pass your liquid, the liquid cannot travel in between the particles, because here the one particle is touching with the other particle. So when that is why whole portion of that solid matrix will not come in the contact with the liquid. So your rate of reaction in this system will decrease to a great extent. This is the major drawback of this pack bed reactor. Now it defines a vessel filled with the catalytic pellets. We can packed with the immobilized enzyme, immobilized microbial cell, and then reacting fluid passing through the void space between them, because this is and pumps can be used to make the fluid move through the pack bed. Application, it has the wastewater treatment process, particularly by using immobilized cells. Immobilized cell system that is used for the production of different products. Particularly we can use high fructose corn syrup by using immobilized enzyme, multiphase reaction, and pharmaceutical industries. Now then we have the trickling filter, which is largely used in the wastewater treatment process. Now major problem with this process, since the particles they resize with each other, as the time increases, the microorganisms that grow on the surface of the solid matrix, and the porosity of the bed will be lost. As the porosity of the bed is lost, then there will be channeling effect. As soon as there will be channeling effect, then the performance of the reactor will drastically go down. So this is the major problem that we have with this trickling filter. So this looks like this. You can see that here your bed, here the solid particles here where the cells are immobilized, and you pass the liquid here is sprinkling. You spread the liquid from the top and take out the liquid from the, when it travels like this, it comes in contact with the organisms, and organisms that utilize the organic matter and convert it into maybe carbon dioxide and cell mass, that and it goes out like this. So your BOD COD value, that whatever contaminants is, that reduced to a great extent. This is largely used in the wastewater treatment processes by different industries and different municipalities. Another very efficient reactor we consider as the fluidized bed reactor, and the reason is that this reactor, why it is efficient, because the particle, you can see that they are not touching each other, they are separated from each other, and here we pass the fluid as a very high velocity, so that the gravitational force, because when we pass fluid at the high velocity, then there will be some kind of axial force, and if the axial force is more than the gravitational force, then what will happen? Then particle will detach from each other, and this is possible when we pass the fluid at the very high velocity. And the problem is that when you pass pass the fluid at a very high velocity. The hydraulic detention time is what? Hydraulic detention time is nothing but volume by volumetric volume of the working volume of the reactor divided by the volumetric flow rate. Now, if the flow rate increases, then what will happen? The hydraulic detention time will be very less. The hydraulic detention time is the time the liquid resides in the reactor, and that is actually the reaction time. And since it is the hydraulic detention time decreases, then what will happen? The reaction time also decreases. You will get very less amount of product formation. So what you have to do? You have to recycle the liquid again and again to get the desired amount of product, because you have to give the sufficient reaction time to get the desired amount of product formation. So if you look at the application of this, that here I want to point out that the typical velocity that we use, that axial velocity of the fluid is varies from 6 to 20 meter per hour, and recycle ratio is very high, 5 to 500, and bed expansion is taking place is 30 to 100%. The expansion of the bed, because you will find easily that how the expansion of the bed take place. Then it is used the multi-phase system with biocatalysts, food processing industries like peas, shrimp and sliced vegetables, etc. is largely used. Then other reactors also we use in the biochemical industries, and what is called bubble column reactor. Particularly if you look at this algal fermentation process, we pass this is the media where the algae grow, and we pass the air, and air gives the carbon dioxide, dissolved carbon dioxide here. This carbon dioxide utilized by the algal cell and fixed in the form of cell mass, and here the gas will go out. Mostly it contains oxygen and unconverted carbon dioxide and nitrogen that present in the air. And this is the air, this is how nitrogen, the liquid is coming in, and is going out like this, and like this algal cell mass production take place. It is largely used for the single cell protein production, and from cheese whey, and also algal cultivation process largely used. Next is the airlift reactor that is used. It is something similar to bubble column reactor, only the different is that there will be inner draft tube. And if you look at the draft tube might be like this, different types. This is the inner draft tube, and here when you sparge air, the air will go out like this and go out like this, but there will be some kind of dragging force here. With the dragging force, some of the air bubble will recycle back to the system this way and this way. This is the two pattern we have. You can have the aeration pattern might be of different type. This is the different internal modes that we have. We can have the aeration here, and then that air bubble will come here at the dragging force, and this is the external circulation system that we have, through which we can have some kind of air bubble that they recycle back to the liquid again, so that your mass transfer in the system that increases. Now, if you look at this is the use where the shear stress in the because you know when we work with kind of living cells, shear force that plays very important role, because if the shear force is very high, that will hinder the growth of the organism. So if the shear force is less, then it will be good for some of the organism to grow, particularly plant cell. And I can give the example of the animal cell. The plant cell, mostly this is recommended that we should grow in the airlift fermenter. We cannot use in the CSTR, where we have the mechanical starter, and when we use the mechanical starter, there will be some kind of shearing effect, and with the shearing effect, the organisms will not grow properly. So it has different applications. In the I told you in the photobiological process is largely used. Also it is used in the plant cell growth, for the scaling up of the plant cell growth is largely used. And next that I want to talk about the different type of miscellaneous reactors. Like we have this membrane reactor, we have we can have the membrane here. You can see this the membrane, and we pass the we can fix our enzyme or your microbial cells here. When you pass your substrate, it will come in contact with this enzymes and cells, and then you can have the desired product at the end. So this is largely used in the monoclonal antibody production. You can see that monoclonal antibody production is also wastewater treatment process is used, also is used for the hydrogenation of the ethanes, that is largely used. Now I want to talk little bit on the construction of material. That is very important, that what are the materials that usually recommended to prepare the reactors. And first characteristics of the material, it should be flexible and it should be durable, because you know that the reactor life should be minimum 10 years, 10 to 20 years we have. So on the basis of that, we should have this the reactor. We should select the construction material accordingly. Then it should be non-toxic to the reactant and the product. This is very important. If it is toxic, then naturally your product formation will be affected. Then resistance to the chemical and metabolic products by the organism. So during the microbial conversion process or during the enzymatic reaction, some product formation is there. So your material of construction should resistance to that, resistance to weathering. This is another, and low cost and availability. This is very important, because cost is a very important factor as far industry is concerned. And most of the biochemical industry, if you look that that construction of material is mostly made of stainless steel, and stainless steel quality depends on the composition of the alloy that we have in that stainless steel. And here I pointed out that if it contains 12% of chromium, that prevent the corrosion, and 8% the nickel, it gives the austenitic structure, which is called smoothness structure of the stainless steel. And even we have seen, if we use the little bit of molybdenum, that further increase the acid resistant property of the stainless steel. So different type of stainless steel that has been used as a material of construction in the industry, in the chemical industry, biochemical industry, different type of stainless. In the home also we use the stainless steel. So different type of stainless steel we use. The high transparency, particularly photobioreactors, this is required. Polycarbonate, I can give the example. This is the transparency is 100%, and we find that light can penetrate very easily, which is very much required for the growth of the algal cell. Now, if you look at this construction of the different the material differ from small scale, pilot scale, and the large scale operation. Because if you look at the small scale, usually we prescribe it should be transparent, so that you can see from outside what is going on inside. In the pilot scale and large scale, not necessarily it can be transparent. It can have, because usually I showed you that this is usually made of stainless steel. And why it's stainless steel? The reason is that if you look at the microbial fermentation process, we have different type of organic acid formation take place, and this organic acid has some corrosive property, and due to this corrosive property, we usually recommend it should be stainless steel. Stainless steel can resist that corrosive property of the or the nature of the fluid. Now we have this different quality of stainless steel. We can have SS304, SS304L, SS316, SS316L, SS317, and for the small scales and big also for pilot scale industries. I work with citric acid industry, and we since we are producing citric acid, and citric acid has a lot of it has quite a light corrosive property, so we usually prescribe SS317 for that. Then stainless steel, I told you 4% chromium that we have in SS304, SS304L and SS316L mild steel and coated with glass epoxy. Epoxy particularly I want to mention in case of mild steel. Suppose in case we use the mild steel, we use the epoxy coating on the surface of the mild steel, so that it makes a barrier between the liquid and the material of construction, and it has the resistant property. Particularly we know the sulfite, if is present in the liquid, that gives some kind of corrosive property, and not in presence of some acidic media. So the epoxy is a very good coating material, which can give a protection to the mild steel material, which so that liquid will not come directly contact with the metal surface. This is largely used by the industry, and it is very useful. And we can see that here that lab scale and commercial scale reactor, how it looks. And a lab scale, I told you that usually we prescribe that it should be transparent, so that from the outside you can see that what is going on inside the reactor. You can have the visual observation like this. But industry, it is not possible. It is a very bigger scale. We operated in a very high scale, that you can see. So here what we do at the top, we have the watch glass. One end we put the light, another end from other glass you can see that what is going on inside the reactor. That is in practice with the industry. So this we'll discuss more in details in my subsequent lectures. Thank you very much.